It's a pleasure to have Lorraine Metskas with us. She's actually a classical musician, or was a classical musician, and then she did her PhD at Yale in molecular biophysics and biochemistry, then a post with John Briggs at EMBL, and then at an MRC. And now, currently, she's a postdoc with Grant Jensen at Caltech, and she's starting a new position at Purdue in biological sciences and chemistry. And I'm told that she's looking for postdocs and graduate students. So if you're in the market, you might want to chat with her, maybe later in the gather town that we have after the meeting. Lorraine, the floor is yours.
Okay, thank you. So I'm going to be telling you today about correlated light and electron microscopy, and specifically looking at practices, challenges, and future applications. So a brief overview of how the talk will go. I'll begin with a practical introduction to Cryo-CLEM, and then I'll move on to recent advances in the field that I've been involved with, particularly parameterization of fluorophores at cryo temperature, and using fluorescence to add new information to the electron micrographs in cases where that would have otherwise not been possible. So beginning with the introduction. Historically, Cryo-CLEM has had one goal, which is to aim the more powerful transmission electron microscope. And so if this is my grid, I have a heterogeneous environment. So this could be a eukaryotic cell, prokaryotic cell, a mixture of proteins, really anything at all that you can freeze onto a grid. And the question is simply, where is my target? And historically, these have been larger targets, things like endosomes or large protein cages inside of eukaryotic cells, or virus capsids purified on grids. And the goal here is simply, if I'm looking for the smiley face, how do I find it if my field of view is only about 1 micron squared? So I could simply use the Krios and spend all of my time taking tomograms of things that are not my target, or I could try to highlight my target in some way. And this is where the fluorescence comes in. So we are harnessing the specificity of fluorescence to couple with the resolution of electron microscopy. So the good news is that this doesn't actually add very much to the workflow. In a normal cryo-EM experiment, you would do your sample preparation, and then you would put it onto a grid and freeze it in the manner of your choice. And then finally, you would do cryo-electron microscopy. With Cryo-CLEM, we only add 2 steps. We have to attach a fluorophore to the sample prior to freezing. This is usually using established protocols because fluorescence has been done for many decades now. And then we add cryo-light microscopy in between the grid deposition and the cryo-EM. And if we were to do fib milling or any other sort of sample thinning procedure, it would happen between the microscopy and the light microscopy and the electron microscopy. So I'm going to walk you through how these correlations work at the microscope and then in the post-processing. On the left will always be the data from the cryo-fluorescence microscope. On the right will always be the data from the electron microscope. In this case, it was Techni F20. And it's important to note that in the cryofluorescence, we actually have 3 different types of data. So we have a bright field or transmitted light sample. This is just to look at what the grid looks like. Then if you use fluorescence fiducials, you have a channel for that as well. And then finally, we have the signal channel that actually highlights the protein or function of interest. So I will take a grid map in the bright field, and I note different squares that I think contain information that I want to take micrographs of. And one of these is circled in red. I then also find other grid squares that can be used as markers. So these typically are things like torn or missing carbon, or some other feature that would be clearly visible, like the center dot on a C-flat. And then I can do a montage in the EM and find those markers and easily find my square of interest. So this doesn't require any sort of computation. It can just be done by eye in a matter of minutes. Once I'm at my square of interest, I can take the bright field information and look at defining features on the grid. So in this case, I'm using empty holes, which have this characteristic white dot in the middle of them in the bright field. And then, of course, they're very visible in the EM. So I can do a grid square montage, find those locations, and put the two maps into registration using built-in cryo-EM functions. Once that's done, I can drop in my signal fluorescence. It has the same registration, so there's no additional computation necessary. And I can simply add these fluorescence points as cryo-EM targets and collect micrographs at those positions. And what's important about the steps I've just shown you is that they are computationally light. There are a number of reasons for this. The first is that our facilities understandably do not want us putting a lot of information on to the computers running these microscopes. They're also very leery about academic software that could be buggy, and they have good reason for that. And also, we have to be aware of the RAM requirements. So if we're uploading high resolution, large images, and then rotating them and flipping them, that could put a strain on the computer. And we want to guard our microscopes. So we keep everything very computationally light, and then we do all the computation in the post-processing. So this has a resolution, or sorry, a precision of about 100 to 200 nanometers is average. So it's enough to aim the field of view. And then in the post-processing, we can get into the tens of nanometers. And so how this works, on the left here, I have my fluorescence image. The green dots are tetraspec beads. So those are my fiducials. And they show up in both the fluorescence and the EM. In the EM, they look like these dark little circles here. And then the red is my signal. So I begin by identifying known positions on the fluorescence and the electron micrograph. So if you're using fiducial beads, you can do either by eye click and point, or you can also do centroid finding or some other method. Another method that's becoming popular is centroid finding on these grid holes. I think fiducials tend to give a little bit better precision still. And then we compute a transformation matrix that minimizes the error between predicted and actual positions on the EM grid. So we're always moving from the fluorescence to the EM. So you stretch and manipulate the fluorescence map and overlay it onto the EM map, not in the other direction. These algorithms, I've put two papers with open source software down here. These algorithms tend to be on the simplistic side, so it's things like least squares minimization for the error, and simple transformation operations. Finally, perhaps the most important step of the post-processing is what we call a leave one out confirmation. How this works is we take one of the fiducial points, for example, this one right here in the center, and we remove it from the calculation. And this allows us to recompute the transformation and then predict where that point would be. And this gives you both a more certain estimate of your actual error, because this calculated error tends to be a little overly optimistic. But more importantly, this gives you spatial knowledge of the error, because you can imagine this grid square here is a 25 square montage. And you can imagine that there might be some stitching issues in there. So in addition to the normal movement of things on the grid due to handling or breakage, we now also have to contend with the fact that there is alignment. And so this gives us the knowledge that everything in this area has a certain degree of precision that might differ from things in another area of the grid. Once we do that post-processing, we create overlay maps. And this is one of the grid that I just showed you. So again, green are fiducial markers, red are areas of interest in fluorescence. And if I zoom in, you can see in higher resolution, here is the fiducial marker underlying the green signal. And here is a fluorescently labeled lipid vesicle. And there are unlabeled ones as well, just as a control. And this is pretty typical of the resolution that we get in the cryofluorescence as well. So currently cryo-CLEM is used primarily as a method still of pointing the electron microscope. And this is well demonstrated in this paper from Elizabeth Wright's group just earlier this year. So here they're looking for fluorescently labeled viruses budding off of the eukaryotic cell. They look at these protrusions and they aim the TEM there, and they take a micrograph. And at this point now, we can clearly see the targets. And so we don't need the fluorescence anymore. the fluorescence aimed the field of view, and now you have an easily recognizable target to do higher magnification work with. And I would say this type of application encompasses about 95% of current cryo-CLEM. There are other use cases, and this is one that was recently published by the Merner Lab. Here, they're using the fluorescence as their primary mode of information, and the EM is playing the supporting role. So they are localizing proteins with fluorescence that they cannot see in their micrographs, and then simply saying, okay, in the tomogram, this area is the area of ribosome exclusion, so we know that these proteins are in a certain part of the cell. What I think both of these cases illustrate is that cryo-CLEM currently is a pick one of two approach. One technique will be dominant, the other will be supporting. And most of the time, as I said, the dominant technique is the one with the higher resolution, which is the electron micrograph. So if we think about the future of cryo-CLEM, where do we think it's going to go? Well, I think that it's going to head in the direction of this use case. So a few years ago, I started a collaboration with the Al Nagar Lab at the University of Southern California, and we wanted to do high-resolution subtomogram averaging to determine structural information about a protein that was thought to form these complex ultrastructures with mixed stoichiometry. So it was a problem that could not be tackled any other way and promised to be a very exciting target. So we collected a lot of tomograms of these outer membrane vesicles, and you can see that they're about 50 nanometers in diameter, and they contain a variety of proteins. And unfortunately, they do not contain the ultrastructure that we expected to see. We know the protein's present, but we have no way of targeting it because we cannot easily, from the EM alone, tell where it is. Now, if I used cryo-CLEM today, it would look like this, because the resolution of the light microscope is poorer than the field of view that I'm showing you. And the precision of the correlation is lower than the diameter of this vesicle. So I could not even say if the protein was inside the vesicle, let alone use it to help me target it for subtomogram averaging. I think that in the next 10 years, we are going to be trying to bring cryo-CLEM to this type of application where we can use it to target single proteins, perhaps not with complete specificity for subtomogram averaging, but at least to get the search space small enough that we can tackle that question with EM algorithms like classification or template matching. So this is a slide that would normally hold an algorithm, but it doesn't exist yet. So methods developers in the audience, please take note, we need you. The future algorithm is probably going to contain the following things. We need to do super resolution in the fluorescence. So in other words, moving from this large point spread function to these smaller ones. And we need super precision. So we need to take our current precision of around 20 to 50 nanometers and bring it down to an area that's reasonable for classification to kind of sort out the rest. So if we look at the super resolution side of things, there's obviously going to be a lot in the optics and experiment design. We have to take a look at our microscopes. There are a number of optical constraints that we're still battling. Fluorophore optimization, this is something I'm going to return to, but basically a lot of fluorophores don't function quite the same way at 77 Kelvin. And finally, devitrification and grid design. So right now, our grids are not very thermally conductive inside of a grid square. That carbon does not have a good way of dissipating heat. And so if we use a high power illumination, we melt our sample, which of course then prevents us from doing any EM at all. And so we are locked into using these very low powers. And you can imagine low power means low signal to noise ratio. So if we could get better thermal conductivity in the grids, we could use higher illumination powers without devitrifying our samples. Another thing we have to tackle is deconvolution. So what this means primarily is single fluorophore. So if I have one fluorophore here, another one here, how do I tell them apart? This is traditionally done through blinking. There are targeted approaches like Palm and Storm that switch the fluorophores on and off, but fluorophores also blink more in cryotemperature. So some techniques are actually using that blinking as a way of doing this deconvolution. So there's still active exploration there. We also have to deal with cellular autofluorescence. It's worse at cryotemperature. And there's a lot more scatter and aberrant light. So we have to optimize some of our algorithms to address these 3 points. And finally, we have to deal with the point spread function. So a fluorophore is typically modeled as a 3-dimensional Gaussian of photons. Photons are single particles of light. And then when you image it with your instrument, it becomes typically sort of this elongated form where you have an axial dimension that is more narrow, radial dimension, pardon me, that's more narrow than your axial dimension. And because we know what this form looks like, this point spread function, we can use that for modeling. And so super resolution techniques will actually predict, okay, I have this point spread function, where is my fluorophore within that? And that's a computational approach to super resolution. Now, the reason that this works at room temperature is because our samples are very well characterized. So we have our objective coming from the bottom, then oil, a glass cover slip, and then finally the sample. Now, these components interact with light in different ways, but they all have set distances. And so these different refractive indices can be combined and still used to calculate this point spread function. In cryo, we do not have this. So we have our objective that is much farther away, and then we have no immersion media. So this is just air. Then we have the ice. Then we have the sample, and then we have metal, which is not something you normally would put into a fluorescence microscope. And so we have this combination of things. They all interact with light differently, and there's no set relationship between them. It's actually varying over the field of view. So at this part of the grid, your ice is thicker, your sample's thicker, there's less air. Over here, you're getting full-on metal reflection. And so over your field of view, this model is no longer correct. And so we're going to have to deal with that in the computational processing. Moving over to the cryo-CLEM side of things, so saying where is the fluorescence coming from inside of the electron micrograph, fiducial markers tend to improve the precision, so we need better fiducials, probably smaller things. Right now, our fiducials are 100 nanometers in diameter, so you can imagine there's a bit of error just in finding the center of that. So they need to be smaller, and they need to get inside of cells. So fiducial markers right now do not go inside eukaryotic cells, and this is a problem particularly for fib milling, where you're going to blast away a lot of material. We need to do better correlation. So right now we do have 3-dimensional correlation, but the correlation algorithms are on the simplistic side. For our current data, this is very reasonable because, quite frankly, the data don't support more heavy processing, but in the future they will. So we are going to have to do a better job with these correlations. And finally, we need to address the fact that we are manipulating our grids. So at the best case scenario, you are taking a grid out of the fluorescence microscope and putting it into the electron microscope. There's going to be breakage, there's going to be wrinkling and movement of the carbon. But in the most extreme case, you're also blasting away most of the sample with a focused ion beam, and we need to be able to account for that. So if we think about how these two sides of the same coin go together, in the super resolution, this is our point spread function, and we're trying to find the fluorophore inside of it. So we're trying to make the point spread function as small as possible and the accuracy of predicting its center as high as possible. On the precision side of the CLEM, we are basically trying to decrease our search radius. So we're trying to say, okay, I have a sample inside of this circle. I want to get the circle as small as possible so that I can predict where it is. And it's important to remember that the true error of cryo-CLEM is the convolution of the two. And so no matter how far we progress with the super resolution, if the precision is poor, we're still not going to be able to target single proteins, and vice versa. So this brings me to the final part of the algorithm that I see in the future, which is a feedback loop between the two. So I think we would get our initial decreased point spread function in the cryo-FM. We would send that to the cryo-CLEM. we would then be able to say what's actually inside of the point spread function. So we'd see the thickness of the grid, whether it's capturing grid bars where they are, send that back to the super resolution, do better processing, and go back and forth. So moving on to some new developments. One of the projects I've been involved in has been parameterizing fluorophores at cryo temperatures. So in the super resolution side of things, it would be included in the optics and design. So this is going to require just a little bit of photochemistry, so bear with me. This is my favorite fluorophore. It's called Alexa 488. And when it fluoresces, it undergoes an electronic transition. So there are delocalized electrons, and they're going to be jumping from the highest occupied molecular orbital to the lowest unoccupied, or the S1. So I shine light. Light is energy. If that energy matches the distance between these orbitals, an electron will jump. It'll stay up in the S1 state for a little bit of time, typically a few nanoseconds, and then it falls down again to the ground state. And when it falls, it has to get rid of that extra energy, which it can do either through heat or through releasing a photon of light. How this looks in a spectrum now, if we are looking at sort of a population density of where photons are either absorbed in the dotted line or released in the solid, these are called excitation and emission spectra, and they have these characteristic appearances. So there's always an energy difference. This is because you always lose a little bit of energy in the excited state, typically through motion. And it always has these shoulders, and these shoulders come from substates that are accessed through motion again. So these are vibrations and rotations that allow a little bit of wiggle room in jumping between these states. So a little bit of freedom in how well that energy has to match. Finally, a fluorophore oftentimes will fall into what's called a triplet state. This is done through intersystem crossing, or a spin flip of the electron. And this is primarily a dark state. So it's an unresponsive dark state of the fluorophore where that electron is trapped and waiting to do a spin flip so that it can return to the S0. And this predominantly is associated with things like quenching, photobleaching. So fluorescence has a ton of applications. I'm not going to go through all of these just to show you what they are. But some of them also involve more than one fluorophore. So we can do multicolor experiments in the localization or in any of these others. And the idea here is that you need fluorophores that are separate enough in their excitation spectra and in their emission spectra that you don't have one wavelength of light illuminating both, for example. But we can also use fluorophores that interact with each other. And this is common for techniques like FRET, Forrester resonant energy transfer. And the idea there is that that interaction is characterized by this overlap integral between the excitation spectrum of one fluorophore and the emission spectrum of the other. So why am I telling all of this, and what is the problem for cryo? Well, the problem for cryo is that we have no movement. And with reduced movement, we see changes in these spectra. So the peaks can narrow, these shoulders can be affected because it's harder to transition between substates. Intersystem crossing rates to the triplet state are altered. We don't have collisions, which are one dominant way of getting out of the triplet state. And the spin flips also seem to be happening a little more slowly. And so what this means is that a fluorophore can spend up to 95% of its time in this dark, unresponsive state. And finally, we have fixed dipole orientations. So when you shine light on a fluorophore, it doesn't just matter the wavelength, it also matters the orientation of the light relative to the molecular dipole of the fluorophore. So at room temperature, everything is tumbling so quickly that this is not typically a problem. At cryo temperature, it will be. So we have to think not only about the wavelength of light, but also keep in mind that the orientation could be an issue as well. So one thing that we're doing is to simply characterize some of these changes. And the idea here is that by releasing these databases, we can help people design their experiments, so choosing fluorophores that either will work together or work independently, and that will be bright. And also, we can help microscope design. So we are assuming right now that fluorophores behave exactly the same way, which means that cryofluorescence microscopes are sold with the same filter sets and illumination wavelengths as they are at room temperature. And so we need to be able to say if this is a good practice or not. So we've been collecting spectra at 77 Kelvin, or liquid nitrogen temperature. And the good news is that many of these shifts are pretty subtle. So they will affect fluorophore interaction. So a technique like FRET would be drastically different. But if you're just using them for localization, it's not as big of an issue. What we do see are two characteristic features. One is a narrowing of the peak, particularly in the emission. And another is that these shoulders can become stronger. And this is probably through vibronic coupling. Unfortunately, longer wavelength fluorophores have a tendency to show stronger effects. The reason this is unfortunate is that these are farther away from the wavelengths that are typically associated with autofluorescence in eukaryotic cells. So given the choice, many of us would prefer to use a red or a blue fluorophore, but these are the ones that are going to be more strongly affected by the temperature. And you can see this in the Alexa Fluor 647. Another thing that we're noticing is solvent effects. So all fluorophores respond to their chemical environment. These responses are often very subtle, especially for modern fluorophores that have been optimized to be more stable and robust. And so here I'm looking at Alexa Fluor 647 again. It's in a phosphate buffer, and I'm simply toggling between 2 amounts of glycerol. And you can see at room temperature, this has almost no effect on the spectrum. At cryo temperature, it has a huge effect. And this is played out in the spectra, it's played out in the lifetime, the quantum yields. And so what this means is that we are going to have a stronger response to chemical environment at cryo temperature than we expect from our room temperature knowledge. And where this gets interesting is that eukaryotic cells have different chemical environments in the different lipid compartments. So this can be either a problem or perhaps even a benefit, depending on how the experiment is designed, but you have to take it into account. So all of these results will be released as a database, open access, in collaboration with Delmic later this year. And hopefully we can use that to help people design their experiments and design their microscopes. So I'm going to finish with some new application development. And this is a topic dear to my heart because my PhD was in fluorescence spectroscopy. So it's really exciting to try to bring some of that into Cryo-CLEM. So the premise of this work is that fluorescence can do more than just localization. So if we think about room temperature applications, we use fluorescence to look at stoichiometry of complexes. We use it to understand binding, diffusion time. We even use it to measure distances. And at cryo temperature, all we're doing is localization. So how do we bring some of these other functions into Cryo-CLEM, where perhaps we can add information to the EM that would not have been possible just from visualization alone? And a use case here that I did was influenza fusion. So here is a typical schematic for how this process works. This is a glycoprotein of influenza, viral membrane, host membrane. You begin with a prefusion state where nothing is really interacting. And then from crystal structures, we know that the glycoprotein flips up, embeds, and then pulls back down through a spring force to bring these membranes together. When the membranes are together, we know that there is a point where the lipids exchange between the outermost leaflets, but they are still separated in their cargo. And so this is called hemifusion, where there's a point where there's lipid exchange, but these are still two separate environments on either end. And then finally, this broadens into a fusion pore where the membranes merge and the contents can mix. So we know a ton of information about these processes. This is something that's been studied for decades, but we still don't know how to put all of the pieces together because we can only see tiny bits of it at a time, and primarily through fluorescence. So during the period of about 2014 to 2017, there were a number of tomography projects done on virus fusion. And the goal was to visualize this process and gain insight into how it worked and the things that might have been invisible to us because we didn't know to ask that question with a fluorophore. This is one of my favorite applications. This was from the Lee Lab at the University of Washington. And the reason I like it is because they actually took the step of characterizing different morphologies and quantifying them. And so they tried to do their best to put numbers onto a highly subjective interpretation. And so they identified morphologies like this extended interface, bridging, and pinching, as they call it. The difficulty is that these morphologies don't look like what we expected them to look like from the kinetic schemes. And so we added more pieces to the puzzle. We still can't put together a movie of how this fusion looks. However, if we think about these fluorescence signals from the kinetics, that is an opportunity for cryo-CLEM, because we could add the fluorescence through the cryo-CLEM to the electron micrograph. And so the Hemifusion stalk is what I decided to focus on. And I'm using what's called an auto-quenching fluorophore. So all this means is that a fluorophore, if it's dilute, it's far apart, it's bright. When the fluorophores are brought together, they become dark. And so at high concentrations, you get very little fluorescence. At low concentrations, it gets bright. And so if I mix this, I put a lot of fluorophore into a lipid vesicle. I add it to influenza, stimulate fusion with the pH, and I will get a slew of things, some non-productive intermediates, some things that never interact. But if I do get fusion, this fluorophore becomes diluted into the viral membrane, and so it becomes brighter. And so we get this burst of fluorescence moving from the onset of fusion through pH drop upward. So I spent quite a while finding one of these assays that would work on a cryo-EM grid. I'm not going to go through it, but you can read my paper if you're interested. But the idea is that I froze the sample at a point where I should have had a lot of fluorescent signal. And I get this beautiful starry night landscape. And on this grid are many things, but the bright things should be the influence of fusion. And if I target these, indeed, I see fusion interactions. So this is a way to find a function on an EM grid. I'm not just finding the virus. The virus is everywhere. I'm only finding the virus that's participating in fusion or hemifusion. But we can go even one step further. So here I'm characterizing the fluorescence from a lot of different samples. So I'm characterizing vesicles and virus-like particles that have pretty much no fluorescence at all. These plots, the rectangle is the full spectrum, the full range of intensities found. The center bar is the median. We also have vesicle clusters. They get a little bit of fluorescence, not a lot. And then the hemifusion is brighter, and multiple fusion events, of course, get brighter because you simply have a lot going on. And so I can go to a grid square where I don't know what's going on. I can take high magnification images, calculate the cryo-CLEM overlay, find the fluorescence intensity at the different position where I took my micrograph, and use that to classify what I was seeing. So I can say with certainty, this was an adhesion reaction, this was a fusion or hemifusion reaction. And so I'm using the fluorescence to actually do a functional classification on the grid. And to give you an idea of the power of this, here is one of my influenza tomograms. And so the virus is this long brush-like thing. Lipid vesicles are these smooth circles. And I want you to look at this and try to argue with yourself which of these sites are fusion or hemifusion versus adhesion or non-productive intermediates. And so we do have a number of interactions. You can see a very nice sort of flat interface there. And there's going to be a pinching interaction as characterized by the Lee lab coming up over here, and then again right here. So if we think about which one of these are fusion, it would be pretty hard to say for sure. But if we use the fluorescence, green are fiducial markers, blue, the color of this background, would have been fusion. This is actually negative control. So there is no fusion happening on this grid. These are all adhesion only. And so this gives us an idea of where to place these different morphologies inside of the kinetic schemes that are based on fluorescence. So the future of cryo-CLEM, I think, is going to be in two directions. I think it's going to be pushing the resolution and precision of the technique so that we can target individual proteins. But I think we also can add a lot more meaning to our micrographs. So we can get information like chemical environment, distance between things that we're not seeing, conformational screening of proteins on grids, kinetics through linking with fluorescence signals used in those experiments. So I did not have time to really go through a lot of the work that's being done. There are a lot of groups working on this and they're doing great jobs. So I wanted to just quickly give a resource slide. Feel free to screenshot this. I particularly recommend this cryo-SOPHI paper because it's just a really nice example of taking advantage of a quirk that happens at cryo temperature, which is the blinking of pretty much all fluorophores. And they're using that to actually improve the resolution. So thanks to all of my collaborators and funding sources. I showed you bits and pieces from a lot of different projects today. And then 2 quick advertisements. The first is that I'm a member of the Jensen Lab. We are better known for tomography, and we do all sorts of tomography from very low resolution cellular work all the way out to high resolution, which is my specialty. And we have recently released an ebook, and by we, I mean our science writer, Catherine. It's a free online resource. It contains over a decade worth of tomography, fully segmented with text descriptions, open access. So please check it out. And then finally, as Roy said in the beginning, I'm hiring. So if you have a background in fluorescence and you're looking for a postdoc, if this appeals to you, please get in touch with me and I'll send you a job advert. And with that, thank you, and I will take questions.